Is it live on YouTube? Okay.
Hello, this is Dr. Samuel. I'm a founder of Ethiopian Pegasus Media. So in this media, we have different programs. We mainly focus lecturing sciences and presenting different research findings, which are mainly conducted in Africa, but also in different parts of the world in different disciplines. So for today, we will have a presenter. His name is Stembridge. And so I have to introduce a little bit about Stembridge. And Stembridge is originally from Cameroon. He's from Cameroon and has his bachelor degree in laboratory science in University of Boya, and he worked at regional hospital called Lakitini in Cameroon. After that, he got an opportunity to do his master's in Europe, and the master's is called Masters in Leading International Vaccinology Education, and which is coordinated by different universities, mainly by University of Antwerp, University of Barcelona, University of and Lyon, and University of Saint-Étienne Institute as well from France. So at least 5 universities are coordinating that program. It is a vaccinology. So after he successfully finished his vaccinology education, he joined his PhD program in Germany, and by now he's doing his PhD in Germany in Hermann's Institute, entitled on vertical transmission of SARS-CoV-2 from mother to fetus. And today he will have a presentation entitled on a paper entitled on maternal fetal immune response in pregnant women infected with SARS-CoV-2. So as we know, coronavirus is an infectious disease which is caused by SARS-CoV-2. And so far, almost 423 million of people having infected with SARS-CoV-2, and almost 5.8 millions are died by COVID. And if women are pregnant and they are really at a high risk of getting a serious ill from COVID-19, especially if they have it COVID-19 in the late their pregnancy, and even their baby could be at risk. So it is highly recommended to be vaccinated against COVID-19 to protect themselves and their baby, especially for the pregnant women. So thank you, Stembridge, for joining us to have a discussion on this interesting topic, and we're really glad to have it. So now the floor is for you to present. So thank you again, Stembridge, for joining for such interesting topic. So the floor is yours.
Yeah, thank you very much, Dr. Samuel. It's a privilege for me to be here to talk to you about this paper that was published not long ago, looking into the SARS-CoV-2 maternal fetal immune responses that are going on. Remember, like you rightly said, SARS-CoV-2 has keep on infecting a lot of people with increasing rise in death toll rate. And but of course, the pregnant women is also an aspect whereby a lot of people want to know more about the vaccination and also the immune response that is going on at the level of the fetal-maternal interface, and of course, what is happening at the level of the offspring, from mothers that are infected with SARS-CoV-2. So I will, of course, present to you a paper, and let me start by sharing my screen. So I think now my screen is visible.
It's visible, yes, you can start.
Okay, so yeah, so thank you. Like I said, I'm going to be talking about the maternal fetal immune responses in pregnant women infected with SARS-CoV-2. So, like Dr. Sam already said, um, it's a very important topic for us to look into because it will really determine a lot of issues that are being circulating now, probably on social media and other media platforms, talking about the issue with regard to pregnant women. Should they be vaccinated or not? And if I thought they should be vaccinated, what are the pros and cons with regard to the fetus? So before even talking about the vaccination, it is important for us to see what is even happening at the level of the immune reaction going on at the interface between the mother and the baby. So, okay, so SARS-CoV-2, as you already know, present with several outcomes, such as people presenting with no symptoms, as well as those presenting with severe or critical disease. And of course, during the initial phase of this pandemic, it was thought that pregnant women are more susceptible to SARS-CoV-2 infection due to the fact that their you know, pregnancy in itself is a state whereby it might predispose them to severe or adverse clinical outcome due to SARS-CoV-2 infection. But of course, as we keep saying in publications that are springing up, that it's not necessarily the case. Publications have been able to portray or show that SARS-CoV-2 infection in pregnant women is not necessarily more severe compared to non-pregnant women. So, but of course, we do know that most pregnant women with SARS-CoV-2 do present with asymptomatic as well as mild symptoms. And then also, recently, pregnant women have been shown to be at risk of high or severe um, disease and also mortality as well as preterm birth. But of course, like I said, this information is also being counteracted with recent publication, suggesting that it's not really that different from non-pregnant women as well. So, most units born to SARS-CoV-2 positive women do test negative, and of course, it was also a point of debate during the initial phase of investigating whether SARS-CoV-2 is vertically transmitted from the mother to the offspring. There were initial preliminary studies that were carried out in order to investigate if this virus can be detected in maybe in the fetus, in order to be able to elucidate that it's these children from SARS-CoV-2 positive women are also positive. But as well as we do now know, most of the units born from SARS-CoV-2 positive women are negative, they test negative, and also units who do test positive do not have severe symptoms. But however, the timing of mother to child transmission is still unclear, whether utero or maybe intrapartum or early postnatal period, there is transmission of the virus from the mother to the offspring. So as a result of that, it was of interest in this paper to try to elucidate the fact or characterize the maternal fetal immune response triggered by SARS-CoV-2 during pregnancy. And of course, like you can see here, it's so as you can see here, so here, this is of course an illustration depicting a pregnant woman, and this is the fetus here. And this area here is known as the fetal maternal interface, whereby you do have the decidual from the mother, and also you have the chorionic villi, which is from the fetus. So the chorionic villi and the decidual of the mother, this is what forms the fetal maternal interface. And of course, this STB is a cystic trophoblast, which serves as the barrier between the fetus and, of course, the decidual interacting with the mother. So, of course, the aim is to see if, of course, there is transmission of SARS-CoV-2, and therefore means that it has to leave the decidual of the mother And then through this canonical receptor, either ACE2 or TMPRSS2, which is a protease, which is able to cleave the virus, and then of course with the virus binding to its ACE2, and then therefore transmitted into the chorionic villi of the fetus. So in this study, they had 23 pregnant women, and out of these 23 pregnant women, 11 were negative for SARS-CoV-2, and then 12 were positive by real-time polymerase chain reaction. So they were positive. So out of these 12 positive, 8 of them were asymptomatic, so they didn't present with symptoms of SARS-CoV-2. And then one of these women had mild symptoms, for example, tachycardia and fever. And then 3 out of these 12 women that tested positive for SARS-CoV-2, we had 3 who presented with severe clinical outcomes, such as to the extent that they required oxygen. And out of these 3 pregnant women who were severely, who presented severely with SARS-CoV-2 symptoms, one of them had to undergo an emergency preterm cesarean section. So the methodology of this study is that they collected, of course, samples from the mother and also cord blood as of the time of delivery, and they did serology to determine IgM and IgG by ELISA, and they also did some flow cytometry, immunophenotyping, where they collected blood from both SARS-CoV-2 positive pregnant women and of course the control, that is pregnant women who were not positive for SARS-CoV-2, and then they also did flow cytometry to characterize the immune cells and also cytokine profile, and then they also did RNA sequencing of the maternal tissue as well as the fetal tissue, such as the amniotic membrane and also the chorionic villi and other parts of the placenta, as well as of the decidual of the mother. And then here they also did a kind of RNA-seq, and also they compared this RNA sequence data from this part of the fetal-maternal interface, characterizes the chorionic amniotic membrane and also this placenta villi, and as well as they did RNA-seq to correlate both from the fetus and as well as from the mother, so to see if SARS-CoV-2 infection actually led to some changes within these women that were positive from those who were negative for SARS-CoV-2. And then finally, they also did a kind of immunohistochemistry, whereby they isolated certain component of the placenta, such as the basal plate and of course the placenta villi, and also the chorioamniotic membrane, in order to detect SARS-CoV-2 to see if it was actually present in the placenta. They did immunohistochemistry, as can be seen here, and then from there they did nucleic acid detection of SARS-CoV-2 by real-time qPCR. So the first and foremost, what they wanted to see if whether the pregnant women with SARS-CoV-2 were able to induce both IgG or IgM immune responses compared to the control. And as can be seen here, you see that with the maternal, the blue color represent control, and of course the orange color, so the one which is more intense, represent women who are positive for SARS-CoV-2, but of course they do have severe clinical outcomes. And the fainted color is represent those positive, but of course had mild symptoms or asymptomatic, let me say so. So this way, those that were asymptomatic, and this include those that had severe outcomes. So as you can see here, and then here is the concentration of the immunoglobulin as determined by ELISA. We can see here that IgM, of course, was higher in the maternal blood of women that were positive for SARS-CoV-2, But of course, you see, there is no one in the, it's very low for women that were negative, of course, which is also uh, makes sense with regard to the fact that, of course, IgM is an immunoglobulin which is being known to be produced during active infection. And if you look at IgG, we see here that the same thing is also true. We saw that the SARS-CoV-2 positive pregnant women, they had higher IgG compared to the controls, of course. And when we look with regard to the neonates or the fetus, when the cord blood was examined, we saw that the IgM was like really low in both control and positive compared to IgG, of course, which makes sense because IgG, of course, from has receptors at the level of the placenta, known as the FC and NFC, FCR, neonatal FC receptor, which is able to bind to IgG and then trans and then incorporated into the placenta, which provides immunoglobulin protection to the fetus. But of course, IgM cannot be able to bind or transported through this receptor because of large molecular weight. So IgM and IgG levels were similar in both severe and asymptomatic pregnant women infected with SARS-CoV-2. And of course, the IgG was increased in the cord blood of neonates born with women infected with SARS-CoV-2. But however, the IgM was undetected, similar to that of the control unit, like I said before. So, which actually indicated that this fetus or the cord blood, when this IgM level was really similar to those of the control, showed that there was no active SARS-CoV-2 infection occurring in the neonate. So, and then another thing that it was important to look at is the cytokine profile. Comparing cytokine profile, with control and women that were infected with SARS-CoV-2, what is actually going on. So they actually collected the cord blood and maternal blood as shown in the method, and then they tried to look for some pro-inflammatory cytokines like IL-8, IL-6, and then anti-inflammatory cytokines like IL-10, and also these are also pro-inflammatory cytokine TNF, interferon gamma. And so this is at the level of the maternal blood and the level of the fetus. So when all of this was examined, so they were able to say that pro-inflammatory cytokine response was actually displayed at the circulation in both pregnant women with SARS-CoV-2 infection and their neonates, because as we can see here, we saw that the IL-8, so the IL-8 was significantly higher in the maternal blood of the women infected with SARS-CoV-2. We can also see that there was IL-10, which was also high, but of course we see IL-18 was significantly higher as well. But these other cytokines were not that significantly higher. But interestingly, when we look at the neonatal blood, we could see that IL-8 was also higher. So consistently in both the maternal and the neonatal blood samples, IL-8 was both increased. So this result simply show that the cytokine response is observed in both the maternal and the neonatal circulation upon SARS-CoV-2 infection, and more especially IL-8. And IL-8, as we might know already, is kind of pro-inflammatory cytokine, and it's also known to be recruited during, it is also known to be necessary for neutrophil activation and recruitment. So the next, what they wanted to see was that publication or result have stipulated or elucidated the fact that pregnant women or people infected with SARS-CoV-2 tend to have lymphopenia, and so it was also necessary for them to look at what is happening at the level of the T-cell response in women infected with SARS-CoV-2 and those that were negative, so at the control. So interestingly, what they saw is that women infected with SARS-CoV-2 actually had, decrease in T cells, decrease in T cells, which was statistically significant. But on the other hand, in the neonate, there was no significant decrease in T cells. So as we see, so there was no significant difference observed in mothers or in the neonate in the total number of general leukocyte subpopulation. But of course, there was a mild difference in neutrophils and monocyte reactive oxygen species production, as was observed in the cord blood of the neonate when they were stimulated with SARS-CoV-2. So But of course, we saw that SARS-CoV-2 positive pregnant women, but not their neonate, had a reduced CD4 T cells. And of course, they also had including central memory T cells and Th1-like cells, as well as CD8 cells. So these cells were reduced. So another interesting point was to carry out RNA sequencing to see what is happening at the level of these sites of the placenta, such as the CAM or the PVB, BP, which is placental villi or basalis plate, the basal plate, or also the choroamniotic membrane. So to do RNA sequences to reveal whether SARS-CoV-2 infection has a probability to perturb maternal and fetal immune responses at this site. So interestingly, it was seen that the choroamniotic membrane actually had higher numbers of differential expressed genes, followed by, of course, we see the macrophage also at 12, and at the level of the neonate, we could see here of the fetal side. So we saw a change in the gene expression, 59 of the stroma. So of course, the other cell types such as maternal monocyte, maternal decidual, the fetal stroma cells, several cells also contributed to differential gene expression observed between SARS-CoV-2 and the control. So, but of course, looking into detail again to sequence what was really happening in these three aspects that were noticed to express differential, to present differential expression genes. So at the level of the maternal, we had already saw before lymphopenia, and of course we also saw macrophage 2, but then at the level of the fetus we saw stromal 1. So a Kyoto encyclopedia of genes and genome pathways revealed that each of these cell types basically contribute to inflammatory response. Cytokine receptor interaction was enriched in the maternal macrophage 2, and also coronal disease was also enriched in the fetal origin through the stromal 1. So if you put all this together, it therefore showed that the placenta from pregnant women who are infected with SARS-CoV-2 was altered in their immune repertoire, and this alteration was mainly seen in the maternal T cells and macrophage 2, and also in the fetal stromal 1. And then also they went further again in this paper to carry out more RNA sequences to see what are some of the shared or distinct immune responses due to SARS-CoV-2 infection. And of course, at the level of the maternal, we could see that some genes, some differentially expressed genes were downregulated and others were upregulated. And so those that were upregulated were genes that had maternal biological process that were involved in maternal biological processes like a complement activation, classical pathway, humoral immune response mediated by circulating immunoglobulins, complement activation, and adaptive immune response. So SARS-CoV-2 infection actually led to the upregulation of these differential expressed genes, but of course also to the downregulation of genes involved in phagocytosis, signaling pathways, both distinct pathways, the TNF signaling pathways, and also cell-cell communication and all these. But interestingly, if you look at the level of the cord blood, what is happening at the level of the immune response generated in the neonates, we could see that the differentially expressed genes that were upregulated are actually involved in defense mechanism to fungus, defense response to bacterium, response to fungus. But what is noteworthy is the fact that genes that were downregulated were actually genes that were involved in regulation of kidney development, in kidney mesenchyme development, in neural retinal development. But of course, these were not really significant, as those that were seen in the downregulation that were necessary in the maternal blood. So also too, they wanted to also do a kind of a correlation in the RNA sequence data from the mother, the maternal blood, and the cord blood, and to see what is happening at the level of these cells, like I said before, that were either upregulated within the maternal immune response, such as the T cells, or such as the T cells, sorry, such as, okay, such as the T cells, and of course as of the macrophage at the maternal side, and also, so this actually showed that SARS-CoV-2 infection differentially impact the transcriptome of the mother and the neonate in such a way that these changes are shared in both placental tissues from these when they were compared. And then also IgG was increased in the cord blood of the neonate born to women with SARS-CoV-2, as we already saw before, which is actually consistent. So, and then next again, what they wanted to see is whether RNA or the virus can be detected in the placentas of infected women. So RNAs of SARS-CoV-2 can be detected in the placenta of pregnant women. So what they did was that they carried out viral SARS-CoV-2 viral sequences were detected in positive controls, such as those from the bronchoalveoli lavages, of course, which we all know that this site as well that the virus has been shown to replicate in. But of course also, but not in the placentas of tissue from women with SARS-CoV-2 infection. So when they didn't see this, they thought of doing immunohistochemistry with placental tissue, having both the spike and the nucleocapsid protein to detect SARS-CoV-2 in the placenta. So they were able to actually detect spike and nucleocapsid proteins in women that were positive for SARS-CoV-2 using immunohistochemistry, and they were called this, and this was known as like positive signals, because as you can see up here, so this were actually this is the placenta villus, this is the basal plate, this is the chorionic amniotic membrane. So this is at the level of the spike protein from SARS-CoV-2, and then this is for the nucleocapsid protein, of course, because like we know from the general structure of the virus, we have the spike protein. So you have the spike protein here, but of course inside we also have the nucleocapsid. So it was of interest for both this spike protein to be detected and as well as the nucleocapsid to be detected as well. So these were the positive controls where we were sure that okay, the virus is present, and then this also is those women that actually produce, they were actually SARS-CoV-2 positive, but they also showed positive signals which were not detected initially during the RNA detection. And then, of course, here we have SARS-CoV-2 positive pregnant women with negative signals, of course, and then these were healthy women, of course, that were not expecting, of course, to have any signal. So this collective put together, we could see that the RT-qPCR and histological data could show that SARS-CoV-2 is not detected in the placenta tissue. The placenta tissue, that is the chorioamniotic membrane of pregnant women with SARS-CoV-2. So therefore, what were some of the conclusions that were drawn from this publication is that SARS-CoV-2 infection in pregnant women is primarily associated with maternal inflammatory immune responses in the circulation and at the maternal fetal interface. Of course, we could see earlier that there were some cytokines that were pro-inflammatory cytokines that were cytokines that were upregulated, so that IL-8, IL-15, and of course this was also IL-10 for the maternal, and then also this IL-8 was still seen in the fetus. So only IgG was detected in the cord blood of the neonate, suggesting that acute fetal infection did not occur. And interestingly, they were able to show that SARS-CoV-2 infected mothers and their neonate had increased level of IL-8 in their circulation. And then SARS-CoV-2 infection had, lymphopenia at the level of the mother, but no significant reduction was seen in the fetus. And then SARS-CoV-2 infection during pregnancy was also associated with humoral and cellular immune responses in the maternal blood, as well as with mild cytokine response in the neonatal circulation. So SARS-CoV-2 was not detected in the placentas, as was seen through the RNA-seq, but of course through immunohistochemistry, there were some samples or some placenta samples that presented with positive signals, but of course, which at the end of the day was not confirmed by this RNA-seq sequencing of both the spike and the nucleocapsid. But of course, there were some limitation of this study, such as that the fact that the study didn't put into consideration the time in which the time in which the pregnant women were admitted. So from labor to delivery, they didn't take that length in time. So therefore means that there is no timeline to infer severity of the disease progression. And also the conclusion should be interpreted with caution from the study, since the number of severe cases is limited. Like we saw, just very small, 3 out of 23 were actually presented with severe case. So due to limited sample size of those that were presented with severe. So from this paper, at least we could see that, yeah, so the virus was not detected in the placenta, meaning that it doesn't really pose a risk to the fetus, as was seen in other infections like toxoplasma or rubella or Zika virus. But of course, we should be able to know that studies are still presently ongoing in order to come up with more concrete evidence with regard to this issue. And you could therefore, as of now, recommend that, yeah, it still doesn't pose a risk, but of course, it will be a good idea of pregnant women to also get vaccinated, because like we saw, the risk of preterm birth. But of course, these people are just focused at the level of the immune response. So it's actually a good idea for them to be vaccinated, because if they are vaccinated, they will be able to generate more of these immunoglobulin G, and which, of course, like we saw in these publications, will be able to be transported to the placenta and then offer a protection to these babies in the future. So I think with that, it will come to the end of this paper presentation. So thank you for your attention.
Thank you, Stambridge, for your nice presentation. It was really informative, and I like the way how you present it. So you tell us a lot of things about the immune response of the SARS-CoV-2 in response to from mother to the neonate as well. So that was really interesting, and we have seen also what are the different cytokines that are producing in response to the SARS-CoV-2, which is also interesting. And in your presentation, you also mentioned Tcell production is also there, and macrophage and T cells are there at a maternal origin, but also at fetal level, there is stromal one is also produced. So that was really interesting presentation, and we know that COVID is really a hot issue right now. And when people are getting pregnant, they really scared of the virus, because they risky for their baby. That's why this is really a hot issue. And thank you, Stambridge, again for joining us for such kind of scientific presentation. With that, maybe I can ask you some questions in general. So can we conclude there is no vertical transmission from mother to fetus once the mother get SARS-CoV-2? Can we say like that based on this paper? Can we say that one? Maybe this is the first question. And the second one is still people when they get pregnant, and still they are scared of the vaccine. So what is your message? Just briefly, can you say a little bit more about that one as well? Because when they get pregnant, obviously their immune system is reducing, and they really scared of the virus. So some of the people, they scare them if you get vaccinated against the COVID, then you may have the virus, and again that can be a risk for baby. So pregnant women are still scared of the virus and scared of the COVID vaccine. So what do you say about that one?
Yeah, thank you, Dr. Samuel, for those questions. So beginning with the first one, yeah, So from this paper, we could say that there is no vertical transmission because they were not able to detect the viral RNA at the level of the placenta. But of course, I wouldn't say, of course, like we saw, one of the limitation of the study was the fact that they had very severe, they had a sample size that was small with regard to the severe cases, women that presented with severe cases. So I think a more conclusive statement about that will still need to be made when, several research have been conducted, maybe on a larger sample size, and also with um, with women presenting with severe clinical outcomes. Then we should be able to say. But as of now, I think it's still too early for us to give a conclusive or a general statement that it is not vertically transmitted. But according to this paper, it wasn't vertically transmitted.
Okay.
So to your second question, of course, vaccination in itself is an approach which is being, it's a kind of a preventive approach of disease prevention, whereby it is administered to healthy people. So you should, of course, know that it comes with a lot of fear and anxiety. And with regard to pregnant women, we all know that during the vaccine trials, this is a very delicate group of participants, wherein they only come into the phase of clinical trial after a lot of data have been gotten from a larger set of adults. But of course, yes, so pregnant women, of course, it's something, some of them are scared with regard to the vaccine, but as of now, some of the data we have gotten from publications show that there is actually no risk in women actually being vaccinated for SARS-CoV-2. Actually, it is even much more important because we saw in this publication that there is a transmission of the IgG from the mother to the fetus in those that were positive for SARS-CoV-2, which therefore means that this is a kind of a passive immunity which is being transmitted to this unit, so that when they are born, they have a kind of background protection against the virus when they interact. If they are able to get the virus, and also if pregnant women are also vaccinated, they will also generate IgA, which is an immunoglobulin, which is very important at the level of the mucosal site, and of course to also provide protection against the virus to the babies during breastfeeding, and of course will also give them a form of double protection coming from the IgA and also from the IgG. So I will highly recommend that they get vaccinated. Yeah, it will be a very positive aspect, and of course there are no risks attached to that with regard to causing abortion or affecting the baby. So no.
Thank you again, Steinbridge, for your response on the two questions. So in general, we can say that there is no vertical transmission. That means if the mother get infected, with SARS-CoV-2, there is no any risk for the baby. That is one conclusion that we come up with this paper. And another point that we also concluded that once the mother is once the mother is get pregnant, there is no any risk if they get the vaccine. So anyone who is pregnant, let they take the vaccine, because vaccine is really important in protecting them for having not to have the infection. So that is another two basic conclusion that we made in today's presentation, and we have seen a lot at the immune response, how it is interacting. That is a detailed part. So I really appreciate again Ayuk for your presentation, and thank you very much. And I hope I will meet you for the next time in another topic. So thank you, Ayuk, for a nice presentation again. So have a nice
You're welcome,
time.
Dr. Samuel.
Goodbye.
Okay.
Okay, bye bye.